and welcome to our first in a series of webinars on best practices in regenerative medicine product and process development, in which we hope to share with you a framework for doing good integrated product development and manufacturing process development, utilizing strong hypothesis-based science, applied research, and bioprocess engineering. Today's focus on Know Your Cost of Goods will demonstrate how to develop and use key economic metrics to follow through to your product's development. These include know your product, as a manufacturing process cannot be optimized until you have good characterization and strong assays for your product specifications. The second, know your cost of goods, is the focus of today's webinar. It is potentially the most strategic area in which process and material decisions today can impact your cost of goods for years to come. Finally, know your manufacturing process, where once you have a strong understanding of both your product and its associated costs, it enables you to build an integrated process and product development plan. This will ultimately allow you to relay to the FDA not only what your product should be, but also that you have a process to make it, the QC analytics to test the success of your executed process, and finally, that you can catch when something goes wrong. Early development work is focused on understanding your target product, what needs it addresses, what it looks like, how it functions, its mode of delivery, and how much is needed. As your product profile comes into view, it will be important to define what a clinical and commercial scale cost model looks like for your product. Also understand what aspects of the process have the largest impact on your cost of goods. This lets the product developer prioritize material and process changes that reduce their cost of goods, while being wary of changes that dramatically increase them. Today's goals are to first discuss why an understanding of your cost of goods should be a strategic focus beginning early on in your product and process development. Early decisions during this stage can impact your product's costs of goods positively or negatively for years to come. Second, we want to keep in mind that product quality is always number one, but your cost of goods should be a close second. Managing your cost of goods throughout product development is key to setting priorities during development and driving towards a sustainable business model. Third, but most important for today, is an overview of how to build and use a process cost model for your product development. This is not necessarily difficult to do, but is still highly strategic and impactful, as some process changes can have major downstream implications, which may require difficult regulatory shifts at later stages. We believe if you are an engineer or responsible for a manufacturing process, you should be doing this, as well as if you manage a technical team, you should ask for this from them. This can help you identify your largest cost drivers as well as hidden costs earlier. As mentioned, this early strategic focus reduces your end product commercialization risk. This is especially true as you shift focus from what to make to how to make it. Ultimately, this allows you to begin to validate your business case to ensure clinical and commercial viability, helps to guide and focus resources and efforts to key strategic areas. Ultimately, this will lead to earlier implementation of products and processes that reduce your end product costs. Cell-based therapeutic products are being developed for a broadening range of target indications, as well as by a growing number of application areas, such as cell therapies, medical devices, cell-derived products such as extracellular vesicles or exosomes, and incorporating multiple technologies such as genetic engineering. As noted in a 2017 survey conducted by ISCT, cell therapy products or cell-based products are frequently developed and produced without incorporating cost considerations into process development, contributing to prohibitively costly products. With shortening timelines and tight budgets, this can be a costly trap to fall into. While the focus most novel products are typically in a very defined IP area that will be unique to each situation, manufacturing the cellular raw materials continues to be a major, if not the major area of expenditures during both product development and downstream clinical implementation and commercialization. This was validated in this survey, showing that cell manufacturing, with materials and labor standing out, having the largest impact on the cost of goods. How cost is measured also evolves during development, with early focus on cost per million cells, shifting to cost per dose, and finally to more market-driven considerations such as price per treatment later on. As this is an evolving change in the metrics, it is easy to see how early decisions made in strategic areas during development will have a lasting impact on the ultimate market viability. At Rooster Bio, we are experts at HMSC and cell manufacturing. Therefore, for this webinar, we'll focus on a large vetted industry cost component, HMSC manufacturing, but the framework outlined is applicable to many product types in the cell therapy space. There are three major steps to building a process cost model. First is to create a process flow diagram. This should be done at a scale similar to an end target output or goal, such as a preclinical study or a phase 1 study. Second is building out your bill of materials, using known data as inputs whenever possible. Finally, once a process flow diagram and bill of materials has been modeled, potential process changes should be identified which have the largest impact in reducing your cost of goods. By doing step 1 and step 2 at relevant scales, with best estimates, your largest cost drivers will start to emerge. A cost model is often difficult to do at this large scale. So starting at current R&D scale and extrapolating out is usually a good place to start as you learn how to make larger scale estimates. We start by identifying our estimated process goals, inputs available to us, and a rough roadmap of where we want to go. These will be used as the starting point for our process flow diagram. First, let's identify what scale our process flow diagram should be targeting. Ask questions about realistic end goals based on your milestones. Is it a preclinical study, a phase 1 clinical trial, or it scales to meet commercial demand? What is your estimated dose? How many doses? Can you estimate or build in estimates of loss during downstream processing? Assumptions are not easy, but as you learn more, you can fill the gaps and gain insights into whether your current platform fits your future goals. As you can see, the lot size needs for a typical phase 1 trial can be up to a fold change different than a phase 2 scale, and multiple folds different than a go-to-market MSC needs for a cell therapy. We have a freely accessible blog available in which we show this work in detail to estimate lot size ranges, as well as the manufacturing platforms which are needed for specific end product targets, and this is a great resource to learn more about this specific exercise. For this webinar, we focus on the following scenario for a typical phase 1 allogeneic cell therapy process. As mentioned, we are focusing on the cell manufacturing portion of the process, but laying out the upstream and downstream components unique to your system is also advisable. We have a novel cell therapy application coming out of our research and development, with work being done at a smaller scale and with primarily research-grade serum-containing raw materials with significant regulatory hurdles. Our goals are to transition to a serum-free HMSC system that will reduce your regulatory burden and have a manufacturing target of 2 to 4 billion cells per lot. The key question that this exercise will hopefully answer is, how do you determine what pieces of your solution, such as specific materials and process steps, to focus on to meet this goal? We start by roughing out our plan of action. To do this, as mentioned, we start from our initial R&D process. We look at the manufacturing platform we are currently using and start to ask questions. To make 100 million cells, we use over 20 T225s flasks. Is this sustainable at lot sizes in the billions? Or should we move to other systems, such as multi-layer vessels, to save incubator space and reduce processing time? What is the breakdown of expenditures during R&D? Do any specific cost areas start to stand out? In this case, labor followed by media seem to be the largest expenditures at our lot size scales that we manufactured for R&D. Then we have an idea of what focus areas to start looking at. We will first identify if there are major cost drivers that can be reduced, followed by a shift towards more regulatory friendly materials and understanding that impact on our cost of goods. Hopefully we can find a solution that ultimately fits our end goal needs, with lot sizes applicable to phase 1 trials, as well as having the quality and clinically relevant cost of goods. For our initial process diagram, we'll use the inputs and insights gained during this R&D phase. Since we need approximately 20-fold the number of cells of our R&D scale, we decide to model seeding with 20 of our R&D 1 million cell working cell banks. We also shift from our T225 culture flask to 10-layer cell stacks. However, this allows us to still use our R&D system parameters per unit of surface area to model out our process flow diagram. For a typical MSC manufacturing process, cells are seeded at approximately 5,000 cells per centimeter squared. They are expanded for approximately 10 days with up to 3 media exchanges during that period. This is necessary to meet our lot size needs, and the HMSCs are expanded 3 times prior to harvest of approximately 2.5 billion cells after 30 days. Typical harvest densities are around 25,000 cells per centimeter squared in this case, and labor rates and media costs are based on industry standards. While these are our estimates, it is important that you use your own assumptions that are best for your situation. Once this process is laid out, we can begin to look at the costs associated with it. To do this, we want to build our bill of materials for this process. There are many areas and expenses associated with bringing a cell therapy to market. These include everything from material and labor costs, overhead and facilities, the costs of failed production runs, to downstream marketing, distribution, testing, etc. Early on, we want to focus on the areas which we can best guess and have the most direct implications into our manufacturing process. These are the direct manufacturing costs, but we should continue to be mindful of the rest, especially hidden costs directly tied to our raw materials or process decisions. While we will not go into every detail, we have broken down our cost of goods to build our bill of materials for each step of our process. We divided these into 3 major subcategories: materials, consisting of our cells, media consumption, reagents, plastics and labware, labor, which we have divided between general labor and media associated labor, and finally, end stage quality testing. Again, these are from our working knowledge and insights, but it is important for you to do the same for yourself. What this now allows us to do is analyze for our largest cost drivers. Here's a breakdown of our expenditures across the 3 subcategories. Unlike the R&D scale, labor has become a lesser component of our overall costs, primarily due to our system improvements such as cell stack incorporation. What we can immediately see is that media use and associated media labor is almost half of our total cost, which is a 25-fold increase over the associated media costs at our R&D scale. We next want to begin to identify key metrics, which can be used throughout the rest of this exercise to bookmark how process changes in our model ultimately impact our cost of goods. The first are 2 simple global metrics comparing the total production cost, in this case around $87,000, and our cost per million cells generated, around $37 per million cells. If dose estimates are known, then it can be broken down accordingly versus cost per million cells. As media has been identified as a key cost driver, we will also track some targeted metrics such as total media consumed, as well as an important industry standard measure of media productivity: millions of cells per liter of media consumed throughout the process. This allows us to compare the impact of process and material changes to specific key areas of productivity within our system. As mentioned, it is important to look for hidden costs as well. For example, as media consumption increases, everything becomes more complex. You need to transport and store it, reconstitute large quantities, warm larger quantities before use. Even disposing and waste management becomes more resource consuming. This is the sort of information that is gained by building your models at a target scale, where downstream costs can start to emerge. The hidden process considerations can impact future company strategy, such as facility, labor, and processing needs. Also, you can focus early on key material decision areas, such as finding CGMP compatible or even better analogous systems and sources for your development materials. At the end of this analysis, focus can be turned to our largest cost drivers, which in this case is media consumption. As a first step, we hypothesized that MSCs at lower density potentially need less media early on during expansion, which can be especially true with a rich supplemented media. As an aside, this can be validated at small scale using our product knowledge, such as cell identity or potency assays for comparability, comparing these process changes in T225 or smaller scale flasks. Ultimately, the question is, can we remove a media exchange from our process? This is a simple process change, and we can go back to our initial process flow diagram and make a simple switch, removing a media exchange during the expansion step. All of our other inputs and estimates can remain the same. We rework our bill of materials to account for this, with the associated reductions in costs for media use and labor. We can immediately see that this process change has reduced our overall costs by 11%, and the total percent of costs for media and media associated labor from 46% of the overall costs to 38%. We see this impact in our overall total cost savings of around $10,000 per lot, and an incremental reduction of approximately $5 per million of our HMSCs produced. Looking into our productivity metrics, we see that this reduction in the total amount of media consumed and the reduction in our total costs of goods are of course due to the reduction in total amount of media consumed, resulting in an increase in media productivity. It is obvious that by reducing the amount of media consumed, that we can have an immediate and valuable impact on our overall cost of goods. But the question remains, how can significant quality-based improvements, the second component of our exercise's goal, impact our cost of goods? Serum-based media, while not unusable in clinical settings, carry a higher regulatory burden. A strong quality-based decision would be to move to a commercial serum or xeno-free system. To do this, we will substitute in an industry-standard serum-free media that can be used with similar results in our established process. While these materials carry an incremental price increase, they also typically carry over a process change as well. This is a necessary addition, the necessity for additional attachment factors during our processing. We've expanded our bill of materials to include the addition of the attachment factors in our material costs in this line here. Well, one thing that we can notice from looking at our bill of materials is that for the initial seeding and expansion, the increase in associated costs with the attachment factor are not as apparent as they are if you model this out to the largest scale. This quality improvement, expectedly, carries over a significant increase in our total costs, impacting our material costs in a more expensive media, as well as the additional attachment factor. This is shown here by our media cost per liter, increasing almost twofold. This results in an increase of almost $50,000 without an increase in cell output, meaning our cost per million cells increased by almost 40%. Sometimes quality needs necessitate increased costs, but beware of expensive media and process changes which do not come with any labor or use productivity improvements, as improvements in quality that hinder commercial viability are not always sustainable. The question becomes, can we maintain the product quality and regulatory profile while improving the process? Removing media exchanges worked initially, and as a potential solution that can be validated again at smaller scales. Looking into the commercially available protocols, we see that we can remove another media exchange from this process. This media and coating pair reduce the expansion window to 8 to 10 days. Again, we lay out our bill of materials to incorporate the removal of another media exchange. We of course do see a reduction in cost of goods by removing more media, with around a $20,000 or 33% decrease in media associated costs. This reduces our total and per million cell costs incrementally. However, it should be noted this is still above our initial cost of goods. Incremental increases in productivity can be achieved with process tweaks or slightly less expensive material decisions, reducing our cost of goods. But the quality-related costs may be commercially prohibitive still. Removing all of the media exchange costs from this process would still result in a total cost of over $90,000. And this may be a reality and a necessary change and process for your system. However, early identification of this cost allows you to seek out systems, materials, and processes that potentially carry increased productivity, and it allows you to do it earlier in your process development, focusing your resources on this investigation, and ultimately enabling key decisions, such as where to focus and what to export and what needs to be done in-house, on what to seek and where to seek industrialized solutions for. Ultimately, we use this strategy to develop our optimal HMSC manufacturing platform. This carries clinical quality with a bioprocessing engineering focus on productivity. Using these two, we developed our HMSC and associated media system. We followed this framework to identify key areas to focus on, and these improvements were made in multiple areas to the manufacturing process that ultimately impacted costs and productivity. For example, we higher volume MSC working cell banks at 10 or 20 million cells per vial reduce the initial costs of the initial seeding working cell banks. A rich media developed to allow for an increased harvest density at the same 90% confluency, as replicating cells have reduced cytoplasmic volume, allowing a higher number of cells to be generated per surface area. A batch culture process, removing the need for media exchanges in 2D, as well as being part of a scalable fed batch bioreactor process, reduced media consumption. Along with developing a system that does not require any attachment factors or surface coatings as part of our process. These were all part of a xeno-free system that we developed using this framework to look at key focus areas and places to reduce costs. The system has analogous products and processes for product development, as well as CGMP manufacturing. From a process standpoint alone, we get to the same endpoint in a reduced number of passages and days. While ultimately our media, Rooster Nourish, has a higher price tag per liter, by laying out the bill of materials and looking into an analysis, we can investigate if this more expensive material and process change have enough productivity savings to ultimately impact our cost of goods. When examining our bill of materials, the attachment factor component has of course been removed. We also now have an 8 to 10 day process versus a 21 to 30 day system, which while not shown would eliminate and impact other costs such as suite time, labor, and other areas. This system bringing material and process improvements at the same quality greatly reduced our overall costs, even though the media itself is slightly more expensive, even than the other commercially available media with attachment factor. Both the overall cost of goods and cost per million cells is lower than the initial starting serum-based system, primarily because these were not designed to scale. These savings are ultimately driven by massive and real productivity increases that can have lasting downstream impacts. Identifying these key areas early enough can greatly increase your chance for success. To summarize our cost of goods analysis, for this example, we identified and reduced major cost drivers first. Secondly, we looked at how material and process quality improvements could impact our cost of goods. Finally, we demonstrated the importance of seeking out solutions that, while on the surface may be more expensive, carry quality and productivity improvements that ultimately reduce our system cost of goods. Well, this was just a basic example. We've seen the importance of this across the industry space unfold in real time. Entering late-stage product and process development or a clinical setting with an inefficient commercial or DIY cell manufacturing solution can and has demonstrated significant losses, significant cost and money influxes to ultimately improve. And this has been a focus of a lot of our development services work, both optimizing and scaling manufacturing processes, ultimately to build in commercial sustainability. We've worked with a growing number of product developers who have industrialized key pieces of their supply chain early, as opposed to waiting until late in their product development. This has allowed them to operate with a more leaner focus on their IP or reallocate resources to broaden the range of therapeutic applications and technologies. This impact is potentially applicable, such as shifting resources to COVID-related studies. This has saved years and hundreds of thousands to millions of dollars versus developing every material and process internally. We believe that an evolution of manufacturing platforms will drive cell manufacturing economics for years to come, continuously bringing down the costs and increasing the supplies to support a growing number of technologies coming to market. We use this framework described here today with a laser focus to develop innovative products and processes that continue to drive productivity increases in an industrialized MSC supply chain, while always maintaining quality #1. This is shown here with our scalable bioreactor HMSC manufacturing processes that have a fold change increase over the 2D processes shown here today. To learn more, contact your Rooster Bio representative or visit our Knowledge Center for more educational information. This includes blog articles on developing manufacturing programs, quality considerations, publication and conference highlights, as well as a library of our technical presentations. Thank you.